Hello, I'm Kenta Kobayashi and first year master student. I belong to the Department of Physics, Chuo University. Today, I'd like to introduce the research of my supervisor, Professor Taguchi, on his behalf. This is the title. First, introduction. Methionine is an essential amino acid. Dietary methionine restriction has been reported to be associated with decreased tumor growth in preclinical studies and extended lifespan in animal models. The mechanism by which methionine restriction inhibits tumor growth while sparing normal cells is not fully understood. In this work, we applied tensor decomposition based feature extraction to gene selection from gene expression profiles of two cell lines of RNA sequencing. Second, method. We employed Tucker decomposition using the higher order singular value decomposition HOSPD algorithm. Tucker decomposition is a type of tensor decomposition. This equation expresses Tucker decomposition, which is decomposing a tensor into one core tensor and several matrices. Xijk often represents the gene expression of ICE gene of this human subject's case tissue. Their mean is centered around 0 and their variance is scaled to 1. G is a core tensor that represents the weight of the product UL1I, UL2J, UL3K to Xijk, and UL1I, UL2J, and UL3K are singular value matrices and orthogonal matrices. An overview of Tucker decomposition is presented in Figure 1. First, singular value vector attributes to UL2J and UL3K were analyzed to identify those of interest. Singular value vectors attributed to UL1I were selected so that the absolute value of G would be maximum among the identified L2 and L3. P-values were computed by assuming that UL1I obeys Gaussian distribution and are adjusted by Benjamin-Hochberg criterion. P-values adjusted less than threshold value were selected and genes were extracted. GSE225945 is gene expression profiling about methionine dependence in melanoma cells measured by RNA-seq. The data for the methionine-dependent melanoma cell line A101D and the methionine-independent melanoma cell line MEWO are obtained. A101D represents a typical cancer cell line that exhibits methionine dependence. MEWO is a methionine-independent cell line. We employed an application on R, TD-based UFE. It is a package of bioconductor. This is the image. Third, HOSPD were performed on the third-order tensor. The tensor with dimensions 66,766 times 8 times 2 was formed in this study. The dimension 2 represents A101D and MEWO. This is the image. And then, the singular vectors were selected. As a result, 8,656 genes were selected. The left one is about J and right one is about K. SVV. Subsequently, enrichment analysis was conducted for the chosen genes. This is X3. Go Biological Process 2023. Finally, conclusion. The application of HOSPD to the tensor derived from GSE 225945, followed by enrichment analysis on the resultant genes, convincingly demonstrates the biological significance of the selected genes. Thus, the effectiveness of this methodology is evident. However, given the somewhat challenging aspects in interpreting the results in this study, exploring experiments with narrower conditions may provide additional insights. Thank you.